is the kind of thing that my brain just kind of gravitates towards, and it's like it's magnetic because it's so fascinating. It kind of bends the world, my worldview a bit.
It does. It is kind of world bending a little bit, really, especially in terms of we don't normally think of plants as having that kind of a relationship with microbes. We think of plants as victims of microbes, but not really managers of microbes and cultivators of microbes.
And it often plays out as a passivity that we view plants having when they're really not passive.
They're not at all. No, not at all. They're really very intelligent. Plants are, and they respond to stimuli in the soil, microbial stimuli, and they attract microbes, and then they internalize those microbes, and then extract nutrients from those microbes. So it's a really, we don't even understand we really only understand the tip of the iceberg there. We don't understand all the details of what's going on in the soil.
Yeah, it's super interesting. When I brought this concept up on social media, it's fascinating to see all the different reactions. But one of my friends who is an ornithologist, who's interested in everything soil and plant related too, he's Stefan Sokobiak. He brought up this book, Survival of Civilization by John D. Haymaker, or Hamaker. I don't know how to say his name, but it talks about how they had a theory that roots would attract microbes and then drain them on their surfaces of the root, of their protoplasm, and then they would have all these shells or skins from these like accumulating over the season. And this was kind of their theory. And I was like trying to explain to him, I was like, well, this is actually like, we're seeing these things now. Professor White has gotten so down there, and he's seen it. And so it's quite different. Whole organisms are going inside, but it seems like we've kind of been trying to figure this out for a long time.
Yeah, I don't think anyone really expected that. Not until that 2010 study of the Australians, Queensland University, Pong Fu Lohan. It's a foreign name. I think it's probably a Thai name, but Pong Fu Lohanihiti. But they actually put an article out in PLOS One, where they labeled yeasts and bacteria, and they fed them to Arabidopsis plants and also the tomato plants, and then they labeled them with GFP, green fluorescent protein, so they could track them, right? And then they tracked them through the plant, into the plant, and then they showed that they disappeared in time. 2 or 3 days later, they would disappear. So they're the ones that actually named it Rhizophagy Cycle. This is Suzanne Schmidt and another group was part of that in Queensland University in Australia.
And was that research done with seedlings as well, or was that with more mature plants?
Well, I think they used both, to be honest.
Whoa.
I think they used both. I think their tendency was to use both. Yeah, but it's actually happening at the growing parts of plants. So it doesn't matter if it's a seedling or if it's a mature plant, because the root tips are all the same. They're all young parts of the plant. So whatever is happening there, and the rhizophagy cycle is actually concentrated right there on the tips. And so older parts of roots don't do it. And in fact, in older roots, those root hairs disappear. So the root hairs are concentrated right in there on the tips. And in fact, we suspect that one of the primary functions of the root hair is to do the rhizophagy cycle, because they're so active in this process. But a number of people have pointed out that Yeah, because plants appear to be plants are doing this. They are smarter than we thought, right? They're smarter than we thought. And they're actually a kind of interesting thing is that Charles Darwin uh, wrote a paper, him and his son, I think it was Francis Darwin, his son, about the intelligence in plants. And he actually proposed that the plant intelligence is at the root, at the root tip of the plant. And it's with that area that the plant, and this goes back to what we were talking about, plants responding to stimuli and in various ways. And so one way they respond, I mean, it's interesting, people forgot about that, but it's an actual paper that they wrote. In fact, I think it was part of a book on plant intelligence. And the theory is the brain is in the root tip. But that's where the microbes are being internalized, right behind that tip. And that's where the plant begins to secrete superoxide onto the microbe, knocks their walls off, the cell walls off the microbes. And then the plant, will cycle those microbes around inside the root cells, because this is happening inside the cell. But technically, it's inside the cell, underneath the cell wall, but not in the cytoplasm. So it's really outside the cytoplasm. But the plant circulates them with cyclosis. So it goes, or cytoplasmic streaming, we call it, going around and around. And at the same time hits them with superoxide. And so what happens is they're being leached of nutrients, and they're also being broken up. So the ones that survive are being replicated. And so that by the time the root hair forms, they're beginning to accumulate inside those hairs, and they're circulated along the hairs on the inside, around and around and around like that. And they really are. I mean, you can visualize them under the microscope. You can see the plant circulating these microbes. And what happens is, if the microbes are present, if they have not been removed, sterilized, then they will accumulate right there at the tip of the root hair in a mass. And at the same time, they're being circulated like this. So every now and then, one of them will be caught by the cyclosis and pulled back in, and then it may be replicated again. And so it continues like that. But if they're there, they accumulate at the tip, and then they stimulate the elongation of those root hairs. And so those root hairs will elongate in spurts, a little bit at a time. And as they elongate, some of those microbes are pushed out of those tips of the root hairs. There's actually little pores that develop in the tip. And so they just kind of, when that root hair elongates, it squirts a little bit out. And we know, because we can see them come out in spurts, we can actually watch this. And so as that root hair continues to elongate, it squirts out more and more of these microbes. So it's in essence repopulating the microbes out in the soil, the microbes that work with it. And we've measured a number of nutrients that come from when these microbes are present. Things like phosphorus and zinc, and in some cases nitrogen, because nitrogen actually comes from the cell walls of the microbes. And it could potentially, there also could be some nitrogen fixation occurring, but we haven't been able to prove that yet.
And the identities of these microbes are, I know that they're classified as endophytic because they're going in and they're, but are they like truly endophytic because they're kind of being drained?
They are. No, they're truly endophytic.
Okay.
Endophyte really just means that a microbe goes into the tissues of the plant and does not cause disease.
Right.
It's not a pathogen. I mean, basically that's it.
Right. Not about persistence.
I mean, in this case,
right.
well, no, it's not about persistence, but some of these do persist in the roots. Many of them will go out you could imagine if they're going in they're being internalized at the root tip. Some of them are going in the cell, but some of them remain in the intercellular spaces. And some of these microbes we've seen will uh, will actually go through the cell wall of the plant into the intercellular spaces of the plant. And so they'll be, lodged in there and they'll actually form little discs inside the root. So they will, we haven't written about this, and probably no one else has mentioned this because they're not looking at it. But they can be in those roots. I mean, for example, bacillus will go in once it goes in those intercellular spaces. It will form endospores and go dormant in there. So they kind of fill this tissue of the plant, in all parts of that plant. And so those endospores are there later. They could germinate later. If, for example, there's a lateral root that forms, a new root will form, and then it can colonize that tissue of the lateral root. So yeah, no, these are actually true endophytes. That's what I study.
Wow.
Yeah. For
So
40 years I've worked on these. Yeah.
So that's so amazing. So did you say intracellular? So are they doing what mitochondria originally did ages ago by entering the cell itself?
Some of them, a few of them can go into the cell itself, and yes, some of them do. Most of them, though, no, don't go into the cytoplasm.
Wow.
There are some that appear to make their way into the nucleus even, and I know it's weird. It's weird, but the ones that, and we think that the nuclear kind of invasion phenomenon is a survival mechanism for the bacteria, because the bacteria, if they remain in the cytoplasm, the cell hits them with a lot of reactive oxygen, and can degrade them. And so, either they're on the outside of the side of the plasma membrane, just underneath the cell wall, or for certain ones, alpha proteobacteria, the same ones that originated mitochondria,
Yeah.
they can go into the nucleus. And yeah, so yeah, and other people have documented that nuclear invasion of certain fungi, like for example, methylobacteria, another endophyte that'll do the same thing, go into the nucleus, which is yeah. But anyways, yeah, there's certain other ones that can do the same thing.
Wow. So something
Yeah,
that occurred to me, is there a pattern to this movement within the roots? Is it like tides, or is it linked to daytime, or is there like a heartbeat almost to these patterns in the roots?
Well, we're trying to figure that out. It's uncertain. Is there a day-night cycle that happens? We don't know. We suspected that at one time, but we're still investigating it. It would make sense that there's a day-night pattern. We're actually trying to figure out what is it that causes the why, because we know when the microbes are present in the root hairs elongate, and so we're trying to figure out what the signal is, and our main hypothesis is that the microbes are secreting nitric oxide, and that triggers the root cell, the root hair to elongate, and that also triggers the ejection from the. Okay, and we can, and my graduate student has documented that the microbes are in fact secreting, or a source of nitric oxide, but we're still trying to sort all of that out, and using things like nitric oxide inhibitors and reactive oxygen inhibitors, things like that, to figure out what exactly is going on. It's kind of it's really interesting, but it's complex to, we haven't figured it all out yet, Matt. That's not a surprise, right? We know that it occurs because we could see it. We've documented it, we've filmed it, we've removed the microbes, we've identified all the microbes. I mean, the ones that we work with, we've identified them. We've put them back into plants where we removed the microbes. put them into different species of plants. We've tried to figure it out, well, what is it? The same microbes in one plant that stimulates that plant could also be inhibitory to other plants. And so we're playing around with it as a potential herbicides, that whole phenomenon where they inhibit potential weeds. So the idea there would be to get a crop plant, get the microbes that stimulate the crop plant and that inhibit competitor weeds. So you don't need really any herbicides of any type. You just have growth promotional microbes that you can use. Yeah, but that's also hard to make it work in nature because there's so many other microbes out there.
Yeah, and then
We call it,
the pH
yeah,
are just swinging.
yeah. We call that endobiome interference. So we even put a term for it, right? That because these other microbes go in, they interfere with the inner workings of the way they go into the endobiome of the other plants and interfere with their development. Yeah, that was the idea there.
So when we're observing all these things, are you watching this in real time with an electron microscope, or have you like set a scene up where you let it run and then you scan the footage? Are you like swimming through the electron
Yeah, no, I'll tell
microscope?
you.
Because I
No, I'll tell
imagine
you
these
how this.
things.
Yeah. No. What we do is we tend to work on the light microscope level. We've done some work with confocal microscopy, but most of our work is with the light microscope. And what we use is something called reactive oxygen staining. And that is, we'll use, for example, stain that will stain either superoxide, which is what the plant is secreting onto the microbe, or hydrogen peroxide, which is a byproduct of the breakdown of superoxide. And when we do that, we can actually visualize the microbes because the area around them will stain. And so we can see what happens is the plant is able to detect the microbes, and then it secretes this reactive oxygen on them. And then when we stain it, because it's secreting reactive oxygen on the microbes, we can then see the microbes. So this reactive oxygen staining is the main type of staining that we do. We also can visualize a lot of these microbes using an iron stain. And that is, the reason that works is because the microbes themselves contain a lot of iron. And so with these iron stains, they'll actually pick up this iron stain. And so it's a stain called ferric cyanide. And so we're able to see the microbes pretty well. And then what we'll also do is we will make movies of these microbes in the root hairs using a system of growing the plant on agarose. So in Petri dishes, plastic Petri dishes, and agarose is just, as you know, gelatin substance. So we put the plants to grow in there with the microbes, and then what we can do is reverse the Petri dish and then examine the roots through the reverse of the Petri dish so we can see then what's happening. So then we're actually visualizing living roots, living root hairs, growing roots with the microbes in them. And those, of course, are not stained at all. So we can see the microbes spinning around inside so we know what's happening. We can also see them being ejected from the tips of the hairs. And we've actually filmed that process as well. And usually when I give talks about the Rhizophagy cycle, I will try to, even though those are highly memory, contain a lot, take a lot of memory space in the presentation, I'll try to show those, and sometimes it crashes the presentation. But it's worth it though, because when you see it, then it's people believe it more. It's hard. It's kind of hard to believe when someone just tells you, "Oh, plants are internalizing these bacteria into their cells." First, they're attracting bacteria from the soil or yeasts as well. They attract them to the root tip. They then internalize them into the root cell. I mean, that right away makes, loses some people right there. They internalize them into the root cell. And then they process them and squirt them back out of the tips of the hairs. I mean, it's kind of unbelievable unless you actually see it yourself. And so when I give presentations, I try to actually go through all the steps and show using bacteria where the shapes are clear, right? Where they, for example, something like some bacteria that will have, say, for example, 4 cells, a tetrahedron, tetrad shape, and then show that the tetrad shape as it goes in, and you go into at the root tip, you see the tetrads. Then what happens is the plant hits it with superoxide, and we can stain the superoxide. You can see because it stains blue with superoxide stain, nitro blue tetrazolium it's called, and then you can see the cell walls fall apart from those microbes, and they become single-celled spherical units from the tetrads break apart, and then you can see them, in the films you can see them rotating and being ejected. We have one very nice tomato movie where we show the microbes being ejected out, and what happens is they're ejected out as protoplasts, and so the tips of the hairs, of the root hairs, get these little tiny pores in them, and only the protoplasts will fit through those pores. They'll just squirt them out through those little pores. The whole bacterium wouldn't fit. So the protoplasts will fit out, and then once they're outside, they're no longer exposed to superoxide, and they reform their cell walls. It's really cool. It's really cool. These plants are doing something we never expected that they're doing. They are internalizing microbes into their root cells, and then they're extracting nutrients from them, and they're injecting them back out into the soil. Totally unexpected. Charles Darwin was right. Plants are very smart, and the root is a critical spot. A critical bit of their capabilities is in the root. And the root tip appears important. We're actually looking at some other, just kind of, one of our ideas about humic substances, humic acids, for example. People use that to stimulate growth. But we think that actually the plant uses humic acid as a signal molecule, as a molecule to detect where the microbes are. Because what happens is when microbes break down plant material, humic substances are released. And what happens to the plant is, what the plant will do, at least in the experiments we've done, is they will then take in more of these microbes. And so you get, the plants then will secrete more. When it detects humic substances, it will secrete more of the exudates. So you see a thicker exudate, that which attracts the microbes. Those microbes will then, more of those microbes will then be taken into the root cells, actually. Root hairs will then be longer. So the plants are responding. And right now we're checking to see to what extent do the plants actually respond to humic substances. And one things we're trying to check now is, can the plants actually grow towards humic substances if they detect tiny amounts of it, which would be another level of response. Not only do they take in more microbes and cultivate more microbes, process more microbes, but they actually can then grow towards the source of those humic substances in order to find more of those microbes. Because the humic substances themselves are not actually nutrients. So anyway, that's the signal molecule hypothesis of humic substances. Yeah.
Going back to the brain is the tip concept, I can't help but think of the Spitzenkorper, the tip of the hyphal tip of mycelium.
Yeah.
And there's this enormous force, this enormous potential, can break through concrete.
Yeah.
But then you've got the complexity of microbacteria actually being in partnership and using mycelium to travel. And when it moves, it can change the growth tip or the spreading tip from this side of the mycelial mat to another area. And what has been said is almost instantaneously. this happens.
Yeah.
Which again seems like a decision is being made. It seems like there's an intelligence there because there's a choice being
there is. Well, of course, people are going to think we're crazy for saying that, but there is this whole argument. There is this whole argument, Matt, that, I mean, I'm sure you've heard of this. It's been going around for a while, that slime molds demonstrate a kind of intelligence that, and there's been a lot made out of that. And it is absolutely correct that that fungi respond to stimuli as effectively as any other organism, and as effectively as slime molds. And I mean, there is a kind of intelligence there that they're exhibiting. They're not just, plants, fungi, slime molds. They're not just, innate, things that are just sitting there. They can respond. So this idea that there is a kind of intelligence in plants and fungi is not crazy. They can respond. They can grow towards something. They can, they're in fact, I mean, as you know, the whole concept of the, let's see if I get it right, the wood wide web, right? Wood wide web.
Right.
The idea that trees in a forest, plants are connected with fungi, communicating through those fungi. I mean, fungi are definitely a part of that, a whole part of that system. And so, yeah, it's part of that intelligence in the forest, right? They're part of that. And plants, I think that plants, we will, in time, I mean, it's discoveries like the Rhizophagy cycle and the way that plants respond to these other stimuli like humic substances, will change the way we think about plants. And that, the days, I think, of treating a plant as a, just a chemical receiving entity, they're drawn to a close. They're drawn to a close, and you can see that. You can see it. You can see it with the rise of these microbiome companies, microbiome agriculture companies that are, a lot of companies now are putting microbes on, bacillus and other kinds of things that you can get commercially. Well, those kind of products they're gonna go up, and some people are gonna figure out, maybe they don't even need those products. They just have to, they grow the plant right, under biodynamic conditions, or they have the right kind of soil. They treat their seeds right. They have to store their seeds properly. Then you're going to have plants that are, have the right microbes on them, soils that are very healthy, and you don't need it. And in fact, you just mess it all up by squirting a lot of nutrients on them. Yeah, I mean, we still do it. Obviously, people still put fertilizers on, but I think that as time goes on, that's going to be, there's going to be a greater appreciation that you don't need that.
Michael Phillips, when I was talking to him about all this, he was telling me that when he plants trees, he doesn't do any compost or anything because he wants the mycorrhizal fungi to go, oh, I got to get to work. And they do. And then later on, you can add things and amend and stuff. But initially you want them to meet, like, it's like with children, right? It's like you want them to like get used to and get used to things without too much stimulus, so they can master it and understand what's there. So
well, and actually some of those microbes, some of those mycorrhizae are already in, like, if it's a soil where trees are growing and have like ectomycorrhizae on them, they're in that soil. And if you put compost there, you're going to create a barrier for those other fungi to make it through to colonize and sometimes bacteria do interfere with mycorrhizal colonization. They actually, there is an interaction, put it that way, there's an interaction there that might affect the colonization of the root by the fungi. Yeah.
Absolutely. So one of the things at the end of your paper that I thought was really exciting was that you suggested that there is future implications for rhizophagy, but that it will require further research. And you've hinted at some of it, but what other
Yeah.
implications does rhizophagy have for gardeners, for farmers, for
Well,
all?
I mean, yeah, good question. Simple things might be like how you treat seeds are really important. You want to maintain the native microbes on seeds. You know, we have I mean, we have this uh, habit or practice to produce seeds, to when those seeds are mature, to take them off the plant and to take them in and store them in a dry place. Well, actually, what happens when you do that is the microbiome doesn't develop on those seeds. There's what we call is an after ripening time, and that is that it takes time out in nature with water, with moisture, with cool and warm temperature kind of variations, splashing from the soil, uh, colonization from other parts of the plant. It takes time for the microbiome to mature and for all the community of microbes that it will colonize, it will be on that seed to get on the seed. And if you store the seed, if you have too much hygiene, those microbes don't develop there. Those microbes are very important for the seedling in terms of its early development. You know, I mean, what happens is you leave the plant, the seedlings, vulnerable to disease. And so one of the things that we did when we started agriculture is we started saving seeds and trying to, store them in places where they were away from moisture and so forth. And that actually reduces the community of microbes on those seeds and makes them more vulnerable to disease. And they also don't develop properly. And, when you put them in the soil, then they don't have the good microbes with them. Yeah. So, I mean, there actually
Holy
was a
cow.
study. Yeah, no, it's really interesting. There was a study of a wild tobacco that some investigators did in Utah, and they took this little wild tobacco, an annual tobacco. This was published, I don't know, 8 years ago, something like that, maybe a little bit longer than that. It published in the Proceedings of the National Academy of Science. And what they did is they took this wild tobacco from nature and then they started cultivating. They put it in cultivation. And each year they would collect the seeds, they would take them and store them to the next year. It was an annual tobacco. And they would plant them again the next year. And, this is in Utah, they were doing this, so it's pretty dry. And what they found is after about 7 years, they found that they started having a disease. It was a wilt disease, and it started wiping out their plants. And then they went and they started looking at the plants, comparing them to the wild plants. And what they found is that the plants that they had cultivated, they were just 7 years cultivation. What they found is that those plants were losing microbes because of the way they were processing, and they were losing microbes. And so they started having this disease. And so what they did was they then took these microbes, certain ones off the wild plants, put them back on their cultivated plants. And voila, their wilt disease was cured. So this is what's happened with agriculture. And in fact, this is gonna blow your mind, actually. This is what happened with corn. And with corn, I mean, we like to think there are biodynamic ways to grow corn and grow it with microbes. But actually, even from the very early days, the Indians, the Cherokee, and the other Indians actually had ways of
Store it
getting
underground.
microbes. Well, they may have done that. They stored it. They probably did store it underground. You know about that, right?
Right.
Well, they also would do this. They would also go out. They called this, the Cherokee called this the corn medicine. Cherokee corn medicine was called. But they would go get wild grasses and things like Phragmites and Hystrix, the bottle brush, and Elymus, the Canada wild rye, and stuff like that. and they would get the roots of those plants, and they would take those plants and they would put them in the roots in the water, and sometimes they'd heat that up, so they get rid of a lot of the bacteria, but they keep bacillus there, and the heating would wake up the bacillus, and then listen to this, Matt. They would then take their corn and they would put it in this water they got from these other roots, and then they would germinate their corn, and then they would take their germinated corn and they would go plant that, and they would have a better effect with their corn, and they would reacquire those microbes. So they were doing, this microbial kind of this microbiology, microbial agriculture, but to them it was magic. This is the magic. This was magic. This was Cherokee corn medicine. You can read about that in. There's a book by an old Cherokee medicine man. Oh my God, I don't have it here. I don't know. It's called the Lonesome Dove or something like that. Something Lonesome Dove Lake or something like that lake. I don't know.
Wow.
I can send it to you afterwards.
So
I wasn't prepared to tell you that story, but
so
you got it anyway.
Well, I am so excited about this, so grateful, because so many of the people that I know are drying their seeds, sometimes out in the sun. They're solarizing them. They're drying them out. It's oxidizing those microbes. And then I have friends who are talking to me about the fact that part of the reason that we don't have the nutrition that we used to have in our plants 50 years ago, 60 years ago, was because we've lost the endophytic microbiology, and it's translated from season to season in the seeds. And so he's all focused on getting it back into the plants and everything. But the linkage between seasons of how we treat our seeds is so incredibly important. I'm a seed person. I used to be a representative for Baker Creek Heirloom Seeds. And we've talked about things like using compost tea to spray down on our seeds before we dry them down. And that's okay in my mind now, but not perfect, because you could be displacing things. And then if you've got a bacterial dominant tea, you could really screw things up. And it's more like the IMOs, the natural farming IMO cultivation, that Cherokee story is so incredible, because that kind of root soak, taking the IMOs from other plants is so rigorous now. I mean, this is what we're doing in order to get mycorrhizal fungi.
And actually, I hate to say it, but what the Cherokees were doing is actually better than what we modern people are doing, because they were getting a whole community of microbes that are adapted to grasses. They were going to these wild grasses. And the other thing they were doing, because they were heating, they were getting rid of a lot of these other bacteria and concentrating on the bacillus, right? So they were getting all these bacilli and putting them back into the roots. And so they got a whole community of bacillus that maybe from different grasses, not just one grass. And whereas we, in our modern methods, of course, I'm as modern as everybody else, takes 1 or 2 or 3 or 4 microbes, right? And use those, when really this wild method, the Cherokees, the Indians, and Cherokees weren't the ones. I mean, they're the ones that we know about, but probably all the corn growing Indians figured out how to do this. Most probably, we don't know everything about it, and most people don't write anything about this, this superstitious stuff, but they were doing, plant microbiology, and it's really cool. So we're working actually with a organic corn breeder named Walter Goldstein, on trying to get some of those microbes back in. And I was just waiting to see if you responded, see if the person you were talking to was Walter. No, it's somebody else though.
No, I was talking to Michael Collins, one of the first farmers for Chez Panisse, and his whole thing was he would go down to the Amazon, and he was looking at what they were doing there to make terra preta, and still the traditions that kind of lent itself to what they used to do, to try to trace that history. what's interesting, the overlap there is they're making chicha, but they're brewing it over low heat in these clay ceramic giant pots essentially, and the
Yeah.
grandmothers are going around spitting in the brews.
Interesting. Wow. Wow. Wow.
Yeah, and so he was telling me all this.
Well,
I'm
they're
like, there's a
getting,
lot of layers.
yeah. Yeah, it's interesting. Yeah, that's really interesting. Well, they must have figured out that that's necessary in some cases. You know, that may be spitting in it after it starts to cool down.
I don't
Maybe
know. I don't know the timing.
to
Great
reinoculate.
question.
Yeah, the timing may be important because they may have killed a lot of the microbes, and then you know you'd have bacillus surviving. But then if they spit in it, they reinoculate it with a lot of other bacteria. Yeah.
Yeah, they may be like
You,
chewing something when they do it as well. I don't know.
they could be, yeah, chewing a root or something like that. You're absolutely right. They may be getting microbes out of other plants by chewing them. Yeah, no, that's interesting what they do. No, I mean, in fact, that might, if they're chewing plant roots and then spitting in them as they heat it up. See, what happens with bacillus is heat activates bacillus endospores. And so, you know, the process that I said about bacillus actually burrowing through the cell walls and going in between the cells and the roots.
Right.
So plants can be filled with these bacillus endospores because of that process. And so the roots, if they're chewing on roots, there's probably a lot of spores there. And then if they're spitting it into some boiling or some hot water, that then activates those spores and germinates them. So they could be essentially pasteurizing whatever it is they're cooking, right? Water plus other nutrients in there, and then putting their endospore mix from their chewed roots in there. I mean, I don't know what they're doing. We'd have to study
Well,
it to see
chicha is
exactly.
corn beer. So chicha is corn beer,
Yeah.
and what they did was over these
Yeah.
pits, they would cook it, and over time, what happened is all these pits basically turned into these fermentation areas because leaky pots, broken pots, they would turn them into compost heaps. And that's kind of the theory behind where Terra Preta came from, because we don't actually know. We've got these observations and we're trying to link them behaviorally,
Okay, I see.
but we
Yeah.
don't really know because
Yeah.
we haven't seen
So
it.
there's a lot of microbes. There's a lot of microbes in that. Those remains, essentially. That's the remains of their fermentation process.
Right.
That's interesting. Yeah, that's interesting.
Yeah, so it's like the corn mash. So they've got they probably have, you know, the cerveza yeast, which is endophytic.
Yeah.
They have a bacillus. And if they're, you know, doing anything with milk, I mean, with lactobacillus, right? Or even grain rice can do that.
You'd have to really get out find out what they've got. You get some samples
That's what everyone's been talking about.
Yeah. Yeah, there's different kinds of yeasts too. There's it may not be all one yeast. It could be a mix of yeast, and it could be bacteria mixed in there too. More than likely, that's what you've got. It's a complex community of microbes that's doing a thing on that in that system. But then, as the alcohol level goes up, a lot of those microbes could be knocked out, and except for some of the yeasts that could survive.
But I think the low heat actually also prevents it from getting too alcoholic.
Oh, really?
I think.
Okay.
Okay. I mean, it's an open top. I mean, I've done, I'm not a drinker, but I know how to make alcohol. And you've got to take it through a first fermentation, and then you have to cap it to get it to that higher level where people get drunk.
Okay.
But yeah, no, it's so fascinating, and their techniques are so. There's such a hidden complexity to half of these techniques where it's like they're spitting, but they may be chewing something, or you know what I mean, or they just drank a tea or something like that, right? It's amazing. I think that the seed insight that you gave is so incredibly valuable I know that there are people suggesting that you can spray flowers to infect the seeds for the next generation, but wow, that's just so incredible. So so you still would keep seeds in a cool, dry, dark place, and then you would use a root soak system to
I would say that's the reality. That's the
yeah.
reality of, you know, we're not going to change how we get seeds, right? But putting microbes back on those seeds would be something that makes a lot of sense. You know, we have really, you know, we've lost the microbial partners to a lot of our crops because of the way we treat seeds. And even to some extent, we've gone, you know, to tissue culture approaches where we produce almost totally sterile plants,
Mm.
and then try to take those. And of course, you know, what we've done is we've made our plants more dependent on agrochemicals, on pesticides, you know, to keep them. Whereas we know from our experiments that we could, if we have the microbes there, we could cultivate, at least in the laboratory under experimental conditions, we can cultivate plants totally without agrochemicals, just using microbes. And the microbes, many of the bacteria, in fact, some of the bacteria will go out away from the, and these is Rhizophagy Cycle bacteria, they will go out into the soil and they will colonize pathogenic fungi, and they'll cause those fungi to leak nutrients, and they become weak. And so they're no longer virulent, they're no longer pathogenic. So these same bacteria can defend the plant from, and basically all they're doing is they're colonizing the fungus, the fungal mycelium, and they're making it leak nutrients. And we know that because the fungi, the bacteria will grow on the surface of the hyphae, and the fungus in the soil won't sporulate in the soil. So basically, we think that using microbes alone, we could cultivate plants. You know, I mean, you know, one crazy vision of the future is we could dispense with all the agrochemicals and use our microbes, for nutrients, use our microbes for disease control, you know, use our microbes for stress tolerance, because certain of these Rhizophagy cycle microbes, when they go in, cause the plant to secrete reactive oxygen, right? That causes the plant then to upregulate oxidative stress tolerance genes. And so the plant then is automatically tolerant to like heat, or, you know, high salt, or other kinds of, even pathogens, other kinds of, because most stress is oxidative stress. And so because these plants are doing Rhizophagy cycle, they're using reactive oxygen to control those microbes, they're more hardy, they're hardier, and more oxidatively stress oxidative resistant than plants that, say, for example, you squirt nitrogen on, or squirt other, you know, fertilizers on. So I mean, it strengthens the plant. Plants become stronger and healthier, and it does affect. We did one experiment using. I had a student using carrots, and it was carrots, and she put microbes on, and with one particular microbe, she found that the chemistry of the carrot was enhanced, and carotenes were produced in, you know, like a double what you get in plants without this particular microbe. So, you know, what happens is, because with certain microbes, it was a bacillus. It was a bacillus microbe. It was a bacillus actually from a different species of I forget the host that it was from. I think it was another carrot family plant, though. It was another carrot family plant. I forget which one it was. I have it in a you know, a PowerPoint presentation here somewhere. But the point is that the whole chemistry of the plant is all altered. So, you know, in terms of taste, you know, these, if you feed plants with microbes versus squirt uh, fertilizer or inorganic fertilizer on the plant, they're going to be totally different. One is going to have a chemical profile that's very different than the other one. And the one that will have most likely that you'll see the richest profile is the one with the microbes. And so where I know there's this big argument, has been for years, where some people say, oh, fertilizer, it doesn't make any difference with the plant or organic.
Mm-hmm.
It actually is, there's a reason that it does. It is logical that it does make a difference. And it's because the plant is responding different. And we know that the plants are oxidatively very different when they have the microbes versus when they don't. We also know they're weaker if you just feed them with more susceptible to disease if you just feed them with fertilizers versus, and the reason is because it's oxidative stress, versus give them microbes. They're hardier plants with microbes.
With corn, I've been trying to replicate the indigenous model of genetic revival or regeneration, because out of Mexico and Peru, the genetic variation of corn has essentially flowed up north towards us. And routinely, I mean, it would come up in waves from these seed savers and exchangers. And so I took Peruvian corn from the highland valleys, Piscaranto and Cuyuchuspi, and I didn't know at the time that no one had been able to do it. But apparently no one had been able to grow it to get full corn ears and be able to save seed. And I just understood the timing of requirements. And so I put it on the dark side of a hill, and it only got 4 hours of direct sunlight. And it was hot, long season in California. And so most people, when they find out that it took May to December to grow, be like, what? But the next year it jumped an entire month earlier. So it adapted really fast.
It was adapting.
Right. And so I was like, this is what literally happened with the Native Americans who brought new seeds in. They would see this gugung adaptation. And this is why corn is corn. And I brought it in because in my mind, the white and the yellow corn that we're all eating, and even, and this, I'm not trying to, talk negatively about anyone, but it's lacking in nutrients. And so to a certain degree, is the genetic variance lacking in a lot of the North American indigenous corns, because only one family was growing it, only one person or a group of families was growing it, holding on to those genetics, which are very precious and very important. But when we do that genetically with a bottleneck, and we're not doing what they do in Peru, which is open pollinating everything and having just wide genetic differences between things, and they literally maintain a lot of their genetic integrity despite being open pollinated. So you have these incredibly vigorous and genetically, I would say, not in a negative way, but dominant, because you'll interact with other corns genetically and it will take over their genome of expression. And so I started growing hardy land races of northern corn varieties alongside Peruvian land races to get them to cross. And my whole thought was, even if I don't get the Peruvians, those genes are going to go onto my shorter season crops if I can time it right. And so what I did was I got the shortest season corn and did it two to three times in a California season, while this one long season corn was going next to it. And I was able to get this transference. But I don't know if we can do that with everything. Because I know I'm getting those. What? Yeah,
Did
I
it
did.
actually cross?
Oh yeah, no,
It
I've
did
gotten it to
cross.
cross.
And
And
you could see the variance in between. You could see the variation in between the two types.
because I
Yeah.
selected a purple
Yeah.
speckled variety, and the purple
Okay.
speckles are just,
Huh.
amazing. And
Sounds
so,
interesting.
I
Sounds
got
interesting.
the genetic
Yeah.
propensity to release and the nitrogen fixing gels, the mucilage, the mucus, coming off those aerial roots that everyone's
Mhm.
raving about now that it has, nitrogen fixing microbes in it. But I have that, and I feel like it's, I mean, and of course, we just discovered it's in sweet potato leaves. It's in sugar cane, nitrogen fixing microbes. I suspect it's in almost everything. And it's because of what's happened, the way we've been saving seeds, like you talked about, and other factors, that we've lost these microbes. And I'm wondering what you would suggest, or what you've seen would be the best way to bring it back. Is it cousins from wild plants, other countries, more genetically diverse land races? What do you think?
I actually like, I mean, yes, I mean, I would normally say, and then I want to say something about your corn and those aerial roots in a minute. But I would say, yeah, it's wild relatives would be the way to go back and get the original microbes from those wild relatives. But I kind of like what the indigenous Americans were doing to get growth promotional microbes back for their on their corn. I think that was a neat strategy, and that you can get that was a really cool thing that we've yet in modern times tried to replicate what those Native Americans did, simply because we didn't know what they were doing. But I mean, that'd be something really well worth trying.
I'm going to do
Um, yeah, you should. I figured you might. Yes, yes, yes. That's a cool. I want to know when you do it, and I can help advise you on it, so we could do it different ways, a couple of different ways, with this different kinds of amounts of heating and stuff like that. I mean, you could turn an invasive plant like Phragmites australis into a cash crop as a source of microbes. I mean,
Wow.
imagine. No longer will we be trying to kill the Phragmites. We try to cultivate it and get those microbes on the roots. Yeah.
So beautiful.
But anyways, like you said, the wild relatives, which is what you've kind of figured, and we've always said that too. Um, that makes sense, and that's what we found. We could do that. With the corn, I want to go back to the corn. It is interesting. We've tooled around with corn a little bit too, as I said. Um, the aerial roots, it's funny that a plant has that. Um, there's one hypothesis about corn is that it evolved actually in the floodplains of riverbanks, and that those aerial roots at the nodes that keep going, they're there because the silting happens, and they keep getting silted up, and so every time it's a new start, right, on those aerial roots.
Yeah.
And so those roots that have the gelatin on them, that's exudate from the root tip. And so they're cultivating microbes for when the soil covers them over. And then they have those microbes there ready to grow and to start internalizing them. And some of those are nitrogen fixers, and they may be fixing there on the tip, but they also may fix internal once they're inside those roots. That is yet to be proven, yet to be demonstrated. But the way corn works is those are not likely aerial roots that are intended by Mother Nature to be aerial.
Right.
Those are likely intended to give the corn plant, that grows over, you said it was like 6 months or 7 months or 8 months. What was it?
Well,
May
May, June,
to
July.
December.
Yeah, it's many months. It's
It's many
7 months.
months.
Yeah, it's 6.5.
As these plants were sitting on that riverbed, they're being silted and silted and silted and silted, by the spring rains or whatever, right? The summer rains, spring rains and summer rains. And so, I mean, that's probably, I mean, we're just, I mean, I'm not an expert on corn, but we have looked at it, we have studied it, and I know there is this idea that corn evolved as a riverbank species, and that they could have been selected by the natives, from those areas, and likely the natives grew them on those areas because those are nice areas for, where there's lots of open land. You'd have to clear a lot. You could just grow your corn there. Anyways, that's my 2 cents. You could take that or leave it. That's my 2 cents regarding the evolution of corn, the development of corn.
It is a fascinating thing. It is absolutely fascinating. I know people are sampling that gel now and using that in their inoculants for other plants to try to transfer it.
Yeah. So some plants will have that, and they're getting it in the like these land races from Mexico, probably because these plants have not been tinkered with the way our plants have, by hybrids and clean. They're not as clean, right? It's not as clean of a system. They're growing in, a lot of these are tropical, so they're growing in tropical environments, so there's lots of moisture around. It's hard to get really dry seeds in those areas. Probably once we get them back here, though, we start, storing them in cool, dry spaces so no microbes can grow, and we'll lose them. This is just how we do stuff. So we have, we're going to have to find another way to get those microbes back.
That's so fascinating. And it just occurs to me again that teosinte was used on the borders of cornfields and milpa farming systems, and they wanted that blessing of the mother of corn, which is, teosinte is a grass that led to corn. And I think that there's that, again, that wild microbiology being right there.
Yeah, good point. Good point. Yeah. Well, there's a lot to study about all of this. I mean, what we can tell is that plants lose their microbes once humans start messing with them and store them, take the seeds the way that, so they lose them. We know that. Experiments, we could show that. Many experiments have shown that. Likely we wouldn't have diseases if as many diseases if we had the full complement of natural microbes. We wouldn't have to worry about as many diseases, simply because a lot of the pathogens, or at least some of the pathogens, I can't say all of them, but many of the pathogens, once those microbes colonize them, they're no longer virulent, so they don't cause disease. And so those microbes play some really important roles in wild plants, and in, I would think in biodynamic farming, they play important roles.
Did you catch that article about the fusarium going into a tomato plant and then being retasked by an endophyte to play nice and be to be a benefit instead of virulent?
That's what we've been saying. That's what we've been saying for a long time. I think I saw that. Yeah, we see the same thing.
Yeah,
we see that. That's exactly what we see. What we see is that many, and in fact, I started to say this, but I didn't complete that thought, that some of these pathogens like fusarium, if they're colonized, they're endophytic and they don't cause disease. Yeah, they're non-pathogenic. They act as an endophyte. We can see them in the tissue, on the tissue. And they're not diseased. They're not causing disease. We can also, for certain plants like a basil, for example, that the seeds have this gelatin matrix on the outside of them that carries microbes, and so long as those microbes are there, the seedlings are resistant to fusarium. And if we remove those microbes with Clorox, surface disinfect them, and you got to really surface disinfect to get rid of all those microbes, then the plants are sitting ducks for fusarium. The fusarium causes disease, kills the plant.
Wow.
kills the seedlings. So the microbes are absolutely critical to the health of the seedling.
Wow. So I guess you probably don't buy your seeds from, some of these distributors like Johnny's, who's like doing seed sterilization and
I would not recommend that unless you're going to have it. I mean, we got to get our seeds somewhere,
right,
right?
right, true.
I tell you what I would not do. I definitely would not get seeds because it's hard to get seeds with all the microbes on them.
Mm,
But I would not get seeds that have a coating of fungicide or fertilizer on them.
mm,
Any coated seeds I would avoid. Any coated seeds because you're that it's a double whammy. You're damaging the microbes on the seeds and you're damaging the microbes in the soil. And it's just not. You don't need to do that. It's a bad idea.
So my recipe for coating the seeds with nitrogen fixing inoculant, is that
That
problematic?
would No, that's not problematic, but it may not be compatible with your plant. I mean, if
Mm,
just I mean, if it's something that gives a growth promotional response, then it's compatible.
right, right.
But if it doesn't, it may be not working because it may just not be colonized in the plant. It may not be compatible.
Right.
I mean, yeah, you can, I mean, this is what we do. We experiment, right? So you have to experiment a lot, to see
Yeah.
what is going to actually work. And we have these great hypotheses, but to get something to work, you have to try it multiple ways and kind of do little experiments and so forth. Yeah, do your own little investigation of it and see what works. And when it works, okay, you got it.
So
You
in
have
it,
to do the same thing. That's what we're doing in the lab. Go
yeah.
ahead. Sorry.
So
Yeah.
speaking of basil and that gelatinous coating, could we kind of create? I mean, I use alginate all the time. I have sodium alginate in the kitchen. I blend it and add it to water.
Yeah.
But if I had like an alginate percentage water, and then sprayed it on them and let that dry on them, would that give it a kind of almost agar-like coating that would allow protection and kind of a housing for it, or would that set things in a different direction?
Well, it really depends on the seed.
Mm,
Uh, the seeds are evolved to work a certain way. I mean, for example, grass seeds, you consider grass like a fescue grass, for example, or wheat, it's the same deal. You have on the surface of the seed, you have other layers. And the microbes typically, now think of fescue grass, some what we call limmas and pallias, but they're dry tissues, leafy tissues that hang on the outer surface. Those tissues are where the microbes are located in that tissue. If you were to take that and put alginate on it, you create an environment that really is not optimal for that grass seed. It's going to be a lot of moisture. You're going to have, the microbes that need to be there need to have the moisture, need to have oxygen, for example, are not going to have it because you've got it coated in this big liquid thing. If you took the seed and dunked it under water, I mean, you'd have the same issue. The oxygen's not, the levels aren't going to be there for the microbes or for the seedling. For basil, I mean, that's different because those seeds are evolved to have this little gelatinous coating on it. It's a thin coating, but well, no, it's actually kind of thick. I mean, you've seen them before; they're kind of thick. Uh, it's evolved. It's adapted to having that kind of a situation, and so I mean, you could try it with the other grasses, with wheat or whatever, but I don't think it would be an ideal situation. Although, if you were to then spray your microbes on the surface of your grass seeds that are dry, I think that would work fine. Spray them on, maybe spray it on with something that sticks, right? And
mm,
then let it dry on there. An endospore mix, for example.
yeah.
A lot of these dry seeds have a lot of bacillus on them, and they're kind of adapted for carrying bacillus, like grasses. uh, basil, that might be a bacillus too, I think, that comes out of that. But anyways, it does matter, this
Yeah.
alginate. Yeah, people use that, by the way. They do use alginate as a carrier for microbes. So you've had the same idea that other people have had, and tried it, and it did work. It does work.
Excellent.
Yeah.
Yeah, so much of this stuff has been thought about by some incredible people, but we're still figuring it out because in order to figure out, we have to be kind of like meticulous, right? Like we have to control so many variables to actually prove that one part.
Yeah, I think that's a good part of it. I think we have our, for a long time we have our concepts for over 100 years, our concept of how plants get nutrients, for example, and they don't have, the old ideas, except for mycorrhizae, and that's even not super, I mean, that's not super old.
Yeah.
Our ideas are that it's chemical, it's all chemical, and that, we have a lot of, there's a lot of inertia there, that everybody knows that chemistry works, that you can squirt these chemicals and they need this for nitrogen to make protein, so you stick nitrogen there. But it's much more complicated to use ecology. And then you don't have everybody, it's easier to use chemicals, you just pay a little money and you get the fertilizer and squirt it on. But microbes are much more complicated, and you've got ecology involved. You've got things that are recent, like the Rhizophagy cycle is relatively recent, just a couple of years now. And not everybody knows about it, and probably, some people just wonder if it's really true. So there's that whole thing, that it's new science. Not everyone has seen it. Not everyone knows about it. There's only a few people working on it. Uh, and some of the, a lot of the science now we have is focused on genomics, and it's less on ecology. And so, ecology is almost soil ecology is almost it's much less than, and certainly less well supported than you would see, some other areas, molecular biology type work or genomics work. And so people think along these other lines, right? And they don't think along uh, ecological lines as much. That's what I think, anyways.
Yeah, but to me it seems so crazy, because it's like, how do you understand the way those? I mean, when I think about things, I think of arbuscular mycorrhizal fungi and the fact that it's like this 70s ever modulating like analog synth, where it's like every time you hit that note, it's a different sound, because it's reacting to everything around it and trying to cue what the next level of succession is from its perspective. And so when I hear that, they're genetically going through things, like I have this friend named Craig, and he's all about this. And he's just constantly genetically testing everything. And I'm like, isn't it kind of like a soup? I mean, if there's that much variation, wouldn't ecology be the only way to make sense of it?
That's what I think. I think that uh, our work in, say, metagenomics is showing us that there is a complex community of microbes out there, and that we need to, somehow dissect that in some meaningful way experimentally, to actually sort out what is. And so, I mean, I think that the experimentation is really the way that we have to get to the bottom of it, and that metagenomics is good, but it's not going to answer all of our questions. That's the bottom line. But you have so much of, everything is kind of focused on this other route. So the real laborious, time-consuming work is going to be all this experimentation that has to be done, and hypothesizing that people have to do. I mean, in order to do an experiment, you have to first really come up with a meaningful hypothesis and then test that, right, experimentally. And not all the hypotheses have been developed. And so I mean, I think that will be sorted out, but it's going to take us much longer to sort out those questions. And Matt, I don't know if you realize this, but I actually am a mycologist by training. This semester, I have 2 mycology courses going simultaneously this fall semester, and these are pandemic courses. These are online, but I have the students doing a lot of independent work in the woods these days in order to make observations on fungi and to identify a lot of these fungi. They have all these exercises. Some of these exercises have to do with actually mycorrhizae. So we've got like for example, digging up. One exercise is digging up plants in the woods, seedlings of oaks in the woods, and identifying the mycorrhizal, the ectomycorrhizae on those roots. So we're exploring these students in these courses are exploring this whole area. And in fact, the rhizophagy cycle, involves bacteria, but it also involves yeasts.
Right.
And it may also involve some other non-yeast fungi. I mean, one fungus that does this is one called Areobasidium, which is called a black yeast, but it has a mycelial stage. So we may have some fungi being internalized into plant roots that are filamentous, and that because what happens is they lose their cell walls and they go in. You can actually see these fungi look very different inside the root cells than the bacteria do. They tend to be larger and sometimes more variable, sometimes almost like cloud shapes in some cases. And they'll oftentimes fill the whole outside of a root cell when they go in. You see, for example, the pigweed amaranthus plants.
Yeah.
Will have these fungi that go into the roots, and so a lot of the roots will have Areobasidium inside those root cells. These black yeasts.
That's so fascinating because so many people categorize amaranth as a non-mycorrhizal.
Well, these fungi go inside those root cells. So, yeah. They're not like mycorrhizae. They're inside that. They're actually inside the cells. Inside the cell. Yeah. Wow. Rather than on the surface of the root. They're internalized. And so in this case, it is the plant, in the rhizophagy cycle, it's the plant that is internalizing these microbes and not the microbes that are colonizing the plant, so the plant is actively taking these microbes in. And so presumably these Areobasidium, these black yeasts, are being taken in to those cells at the root tip. And so we know that because we could take that same black yeast and we can put it in some other plants and get them to go inside those other plants and those seedlings of those other species as well. So we know that we can get them to internal into the cells of other species. It's just that they tend to inhibit those other species. It tends to be inhibitory to the growth of those other species. Wow.
That's incredible. Yeah, I'm friends with William Padilla Brown, Trad Cotter, Paul Stamets. We're talking about having him speak at my conference this winter. Mycology is a big. I'm also good friends with Peter McCoy, Radical Mycology. He's a good friend of mine. So I am absolutely enamored with mycology. So, and I feel like you kind of have to have, You know, all these different groups and terms that we've created separation around. It feels like now we're going and showing how they are deeply interconnected and have all this crossover genetically or interplay. And if you're not kind of looking laterally, it feels very hard to grasp what's actually going on. Because you're like, oh, but then there's this, and oh, but then there's that. And so, man, it's so awesome that you're a mycologist too, because I feel
Yeah.
very, very, very sure that we have to be lateral like that. And it's excellent that you're
So
doing that.
so the reason that in my lab we might pick up something like the Rhizophagy cycle is because for 40 years we've been studying endophytic microbes. And at first we only studied the fungi
Hmm.
that were endophytes. And that studying involved actual microscopic examination of those fungi. So at first we were looking, I mean, my dissertation, when I was an undergraduate, in fact, it was on the tall fescue fungal endophyte in tall fescue. And then when I did my PhD, it was the fungal endophytes in wild grasses. And so I studied the whole
Wow.
life cycle. They were all fungal endophytes, right, in grasses. And then I expanded at Rutgers, came here 25 years ago, and probably about 10 years ago, I expanded to start looking at the bacteria that go in. But in order to see those bacteria, we had to evolve or develop, invent essentially a new stain, a new way to look at them. So we invented this reactive oxygen stain. And so that became the special lens, the lenses through which we could then see these microbes in the plant. Otherwise, they're invisible. You have to be able to see them. You have to be able to see them. And so we started to see them then. And so then we started to see them. We started to study them. We started to see how they pass through the plants, where they enter in the plants. And so we worked that cycle out. And so then we looked at seedling after seedling after seedling after seedling, looking for these microbes that are being internalized into root cells. And so, but it took that special, endophyte perspective, coming first from fungal endophytes, right? And going through, to bacterial endophytes and studying this whole kind of thing. So nobody else is doing that, right? And so why is it no one else sees it? Because there are no other endophyte people who are doing that. And most of the other people studying soil microbes are not doing that. So
Yeah.
they're not seeing any of that. They don't know how. They don't know how. They've never heard about it.
I've got my microscope right here with me, and I literally, it's so frustrating. I feel like I need special glasses because there's no stain. Everything's invisible, and I gotta look.
Yeah, everything
really
is invisible. So I don't see anything. You see, so that's the whole world. You see, that's the world.
Wow.
Until somebody first sees it, it's invisible. And that's how it is with anything. I tell my students, my students to go looking for fungi now. They don't know, they can't find a fungus, but then when they see it the first time, then they see it everywhere. They've never seen a maitake before, right? Never seen a maitake, a Griffola frondosa. Never seen that before. What are you talking about? It's a big fungus. Go in the woods, they never find it. Then find it the first time. Then they can find it faster than I can after that. So they can see it at 100 feet through the woods. But it's the same thing with science. You don't see it until you see it the first time for yourself, and then you see it, and so that's the way the world is with, any phenomenon that we see with the Rhizophagy cycle. People are seeing it and believing it, but it'll take time for everybody to see it and believe it. So what I do when I go around talking is I try to show everybody what I'm seeing, so that seeing is believing, right? And, hopefully some of those, the regenerative agriculture people and organic agriculture people and even the companies, that deal with growth promotional microbes, many of those, see it and they do believe it,
Mhm.
but it's coming, it's slow, starting with that population going out, and hopefully in time, everyone will understand what's going on in plants. And that's my goal, is to try to spread the word about what plants are doing with microbes,
Well, it's
and
so
in the
critically
meantime study
important.
that.
Yeah, well, it's so critically important, and thank you. If
Yeah,
it,
you're welcome, man. My pleasure.
and
Yeah,
I'll say it for everybody,
yeah.
who's watching. Thank you, because um, we are in a whole society where we can't see it. We're in a state where a lot of things we can't see,
That's right.
and it's this process of elucidation, of seeing things for what they really are, or seeing things for what they really aren't, and having the humility to explore things with that unknowing. Man, well, I am so excited because I know so many folks who I work with who are looking through microscopes, who are going to probably be messaging you.
And
Probably
that's
want to
fine.
be taking your
I'll tell them how
classes.
to do it.
Yeah,
Yeah,
yeah.
yeah. And
And
these classes are online now. But
yeah,
the problem is Rutgers tuition is pretty expensive. I would like to say one comment now about plants. I
sure.
mean, what all this stuff is telling us is that Charles Darwin was, and his son were probably right. And that is that plants are a lot smarter and intelligent than we gave them credit for. And this rhizophagy cycle, internalizing microbes and attracting them and getting nutrients from them, and then repopulating them out into the soil, essentially cultivating microbes or farming microbes, is part of that intelligence. Their plants are, they have their own kind of intelligence, and we're just learning about that. And so to us, that old concept of plants is just these innate things that grow in one place and can't get away and don't think and don't respond, is wrong. You know, it's arrogant of us to think that. They actually it's ignorant on our part to think that. Charles Darwin and his son were probably right. They're intelligent organisms.
I'm 100% there, and I feel like that fungi are right there alongside them as being incredibly intelligent and having the ability to teach us a lot about the way the world works, the way that we can work, especially with fungi, the way the mycelial relationships are, mitigation and facilitation, and it's amazing.
It is amazing. It really is. And here's the added wrinkle for you. Uh, Fungi actually are also internally colonized by bacteria,
Right.
and tends to be more with um, ascomycetes and with zygomycetes. And you've heard about the bacterial endophytes, or uh, internal colonists of like zygomycetes, the VA mycorrhizae, the get bacteria that go inside them. And then you get ascomycetes that actually get a lot of bacteria also that internally colonize them. And we can also see those through the microscope. With basidiomycetes, you do get some colonization, but they actually have fewer, we think, internal colonizers, because they have evolved plugs in their septa. And we think these septa actually prevent this internal movement of bacteria. So when you look at the zygomycetes, you've got no septa there. So bacteria can get in and slide all around all over the hyphae. So they get colonized a lot internally. Ascomycetes, you have the septa that goes in like this, but have a hole in the middle, right? So the bacteria, once they get in, they can actually go less easy because of that septum that goes down there, but they can still get through that plug. But then you have the Basidio, I mean, that was Ascomycetes with the hole there. Then you have the Basidiomycetes with the plug in there, the septum that gets a plug in it, and you can't get anything through that plug, the septal plug. And so the bacteria cannot then move, so they don't readily move. So they are less prone to these, and I'm going to put endophytes or endoparasites than the Zygomycetes and the Ascomycetes. So we still have this internalization phenomenon occurring with those other, with fungi.
So is that micro or proteobacteria? Because it would have to be really small.
Both kind. Yeah, both kind.
Wow.
Yeah, both kind. You can see them. We can see them coming out. We can see them. Well, we see them coming out. We've actually seen them going in too. At the tips, at the hyphal tips, almost like at the root tips, the hyphal tips.
Wow.
Yeah. So this internal colonization is not limited to plants. It also happens with fungi, and that's the only point I wanted to make here, is that you have these bacteria. Now, are the fungi managed in the same way that the plants are? I don't know about that. They could be more parasitic than the fungus. I don't know. I mean, it may be that's the whole reason that septa evolved and got so sophisticated to prevent them from moving in the mycelium. Don't forget, this whole process was occurring in the evolution of eukaryotes, in the evolution of the mitochondria, right? I mean, this is how the mitochondria got there, is you got this internal colonization. And at some point, the microbe was no longer degraded internally. And so then you have what remains is a mitochondria or the structure that evolved into the mitochondria. So this was a process that's very ancient in terms of eukaryotes.
Evolutionarily speaking, was Ascomycetes before Basidiomycetes?
We don't know for sure.
We don't know for sure.
I think so, but we don't know for sure. I don't know for sure.
Well, because Basidio is all the mushroom forming, and then Asco is the one that's more like diffuse, right? And that's the one that's in water as well as in the soil.
Smaller. Well, so here is the logical thing that you can take. Okay, so if you just look at the septa, okay, so you have the chytrids, which are the most ancient in that line of true fungi, right? Chytridiomycetes. That's the aquatic ones. Okay, and then you have the Zygomycetes and the glomeromycota, right? The VA mycorrhizae and those fungi. And some of the names are changing there, but those don't have septa, cannot have septa, right? So those are the ones where the bacteria could get in and slide around. So getting all these endoparasites, we'll call them, limits the whole fruiting body that you could form and the how long the mycelium can live, right? So that's a limitation because you could get loaded with parasites. Okay, so if you then develop a septum where you could limit that, I mean, because they go in and they get underneath the cell wall and slide around in there, outside the membrane. If you then create a invagination that goes in a septum, an Ascomycete septum, that invagination goes in, then there's a block. And so they enter a cell and then they can't go very far unless they can go through that pore, go around and go through the pore. So it slows them down, right? Okay, so what the Basidiomycetes did, that's a development beyond that kind of an open septum structure, is they developed a pore, a proteinaceous type molecular kind of a structure that would go into that plug, into that hole, that pore, and plug it up. And that prevents anything like these bacteria from sliding between those cells. And so we think that was the reason for that pore, that plug, was to prevent finally from any of those bacteria getting into the mycelium and spreading around. And that would enable then these fungi to be very long-lived, right? And to have these big fruiting bodies that they develop, and mycelium that will grow for years and years and years under the soil. And so you get these huge, humongous mushrooms that, you know, that are the same type, as big as a whale or something, you know, because the mycelium is really old, and they could limit these. That's our hypothesis anyway, that limits this internal colonization of this mycelium by these bacteria. And so, I mean, it's not, you know, it's basically an extension of the same process that gave rise to mitochondria in the early eukaryotic cells. It's just that process continued, and they, you know, they limited them rather than turning them into an organelle, right? So
Very,
yeah,
very
that's
fascinating.
yeah.
Wow.
a lot of theoretical stuff there, but you know, I mean, it's we've been studying this thing for years, you know, and so, you know, we would develop some of these ideas. You know, proving those ideas takes more time than developing the ideas, but that's the hypothesis, right? Those are the hypotheses that we develop, and it all hinges on this idea of endophytism and internal colonization of these other organisms, right? So yeah, so all of this comes out of that, you know, that mindset, so to speak.
It is really, really incredible. I appreciate you so much for coming on here and sharing so much knowledge and correcting me in places and helping me explore new areas. I'm gonna go back and massage things a little bit, especially about the amaranth being endophytically mycorrhizal. Does that mean that like beets and brassicas and all the quote non-mycorrhizal fungi, non-mycorrhizal plants, are they all like endophytically fungal?
Yeah, yes, yes,
Mm.
yes. They have fungal endophytes and they have bacterial endophytes. And I mean, what you should think about is the rhizophagy cycle does some of the same things that mycorrhizae do, but they do it at the root tips, right? So with the root tips, you have these microbes internally colonizing, going around, cycling. But with older parts of the plant, you have these other fungi connecting. And the mycorrhizae will connect. The VA mycorrhizae will connect, or ectomycorrhizae will connect. And you know, when the root is covered by ectomycorrhizae, you no longer have rhizophagy cycle happening. Rhizophagy cycle is really a phenomenon of root tips, and all seedlings do it. But if you look at a tree, a big tree that's fully developed, it's got mycorrhizal roots. So those mycorrhizae actually go out into the soil, and they do a lot of that absorption. So they'll absorb, you know, phosphorus and zinc and some nitrogen and bring it back to the plant. But in lieu of that, in things like grasses and things with lots of root tips and lots of fibrous roots, they're doing rhizophagy cycle like mad. They'll do VA mycorrhizae, but I think probably, I mean, I don't know. I mean, a question is yet to be answered. You know, what provides more nutrients? Is it rhizophagy cycle, or is it mycorrhizae? And does it depend on the plants? It depends on the plant. It very much depends on the plant. And so, you know, these are all systems that plants use to get nutrients. And so that's the bottom line. And rhizophagy cycle is one of those systems. Mycorrhizae are other systems. Nitrogen fixing rhizobia is an entirely another system for acquisition of nutrients. And yeah, so anyways, there you have it. And endophytes, you know, some of them are involved in this nutrient acquisition. Others are involved in defense from disease agents, in stress tolerance, in enhancing stress tolerance. You know, we know from a lot of work done on the fungal endophytes of grasses that stress tolerance is a big, big effect. But we also know that certain endophytes in aerial parts of plant will also trigger the symbiosis in the roots, cause roots, for example, with the grasses that have endophytes in the aerial parts, in the leaves, will trigger the roots to then secrete more exudate and take more microbes into the roots. So you have one symbiosis in the leaves triggering another symbiosis in the roots. So you have these plants, you know, these interacting symbioses in plants, and so plants taking advantage of, you know, defense against insects and stress tolerance with the fungal endophytes in the leaves and the shoots, and then getting more nutrients and with the bacteria and the rhizophagy cycle in the roots. And you know, this is just the plant system. You know, they're doing everything. You know, I mean, it really is amazing when you think about it. Interacting symbioses. What we see, what you see actually when grasses will have the fungal endophytes in the epicloid, we call them epicloid, they're ascomycetes, in the shoots, in the leaves. What happens is the plants are bigger. The plants grow bigger than when they don't. And the reason is because the one symbiosis will trigger the other symbiosis, which will then make the roots bigger and make the plant get more nutrients and allow the plant to get more nutrients and grow bigger. So it's but you have to, you know, if you're studying only fungal endophytes, you don't see that. You got to study the fungal endophytes and you got to study the bacterial endophytes in the roots in order to see that. You got to look at both to see what's going on. And scientists, we tend to look at, you know, one thing. And I know we're out of time, Matt. You got me going. I'm so sorry.
Oh no, it's
I really appreciate it.
This is perfect. I really think that what we've covered and all the spaces that we explored, I mean, it's going to make my book better. I'm going to go in and add some of these things into it. And I think everyone's going to appreciate it. I think it was really, really good. I think I'm going to be writing you because I'm going to be watching this because there's a lot. This, like you said, this is the space where we are developing new hypotheses, and the uncharted territory of it is so thrilling. I mean, it is really thrilling. I didn't know about endophytes until pretty darn recently. I mean, it's just not something that is in any of the gardening literature or even any of the, you know, the farming books that you read, and you really just, but it's amazing though, when you just take a step into the published journals, how it's literally everywhere. And half the time they're so zoomed in on this one little thing that you're like, well, I don't even know what this means. And you have to step back and learn about that process, even to begin to understand that. So it's been a long back and forth, you know, of study for me, just getting kind of my bearings so that I could develop a fluency in this space. But with work like yours and with podcasts like this, we're definitely going to be able to spread that fluency further and further. So thank you so much.
Thank you, Matt. Thank you.
All right. Well, I am just so grateful. We'll edit this down, and I'll put bumpers on the front introducing you, and I'll put links to your Regen Ag course with John and all that stuff.
Thank you, Matt.
You're so welcome.
You have a wonderful night. I should say I've also looked at your one of your YouTube videos, I think with growing amaranth, the orange giant amaranth. Remember that?
Oh yeah, I was a young man. I was a young man when I did that.
You were young. Well, we all were. Yeah, you were young then. Yes. Well, you're still young. You're still young. Yeah, I'm getting a bit older though. You know, when I say I've been working on these things for 40 years, you know, you can get a perspective after 40 years of working on this stuff.
Yeah.
Right. 40 years of working on endophytes. So you develop some ideas, at least, put it that way.
Yeah. Yeah. And I look back at some of those videos and I'm just so like, should this be on the internet or should I be taking this down?
I liked it. I liked it. I saw that after I was introduced to you by something I think I saw on where was it? At Facebook, I think. Maybe it was on your Facebook page. And then I saw your YouTube video. So yeah. That was I liked that video.
Okay, good.
Yeah.
Awesome. Well, I'll let you go and have a wonderful night. And thank you so much.
Thank you. Thank you. You too. You too. Night night.
Have a good night. I can't even believe it. The information he covers, how amazing a person he is, the wisdom, the patience. I'm just so blown away. I feel like I should go enroll in Rutgers or get hired there maybe. But I just find this interview to be one of the most fascinating that I ever had the pleasure of doing. And if you want to learn more about this, you want to see the visual for this, you want to read about this in depth, you can go right to James's paper that's published online. It's free. You can read it, all the technical. If you want to read about it in my book, you want to see the visual I created, it looks like this. And if you would like to buy or pre-order the book, you can. It's almost out. The ebooks will be out within the week. Within a few weeks, we're going to have the print happening. We're going to be having the print shipped to me like a week or two after that. We're going to be having then a week or two after that, me shipping it all the way to everyone all over the world. So this is the process. We're in the middle of it. If you'd like to support my work, if you would like to check out my books, because a lot of them are free. I mean, these are Kickstarted gifts to me, so I give them back to the world. They're on the permaculturestudent.com. You can download a bunch. And if you want to support and get into regenerative soil, now's the time to do it. Go support that book, go pre-order that book, and get your copy reserved. This is a limited print run that I'm doing. It will come out at a later point in time online through print on demand like Amazon and stuff like that. But as you know, print on demand, after they run out of those first 50, there's a lag of 1 week or 3 weeks, and then there's another 50, and it's a little wonky. But when I do these orders, I order in bulk, and so you're going to want to get your books now and get your copy reserved, and I'll be signing all the copies that get pre-ordered on my website. So an added extra bonus. Check it out. I am Matt Powers. Grow abundantly, learn daily, and live regeneratively. And build soil, remediate soil, regenerate soil with the new book, Regenerative Soil. Out soon. Go to thepermaculturestudent.com and pre-order your copy. Thank you so much. Have a wonderful day.